It is now my great pleasure to introduce the 2015 recipient of the American Diabetes Association's Banting Medal for Scientific Achievement, Dr. Philip Scheer. The Banting Medal recognizes significant long-term contributions to the understanding, treatment, and prevention of diabetes. Currently a professor and director of the Touchstone Diabetes Center at the University of Texas Southwestern Medical Center in Dallas, Dr. Scheer is known for the discovery of adiponectin and for defining its physiological roles. His seminal work has demonstrated an initially protective role for obesity, keeping excess lipid in adipocytes and out of the non-adipocyte tissues, which after all are not designed to store fat. Dr. Scheer's discoveries, combined with the innovative methods he developed to study the individual contributions of specific cells to the regulation of metabolism, have indeed defined him as one of the most noteworthy and creative scientists in the field. Dr. Scheer will now present his Banting lecture titled "The Multifaceted Roles of Adipose Tissue, Therapeutic Targets for Diabetes and Beyond." Dr. Scheer?
Thank you. Thank you. Thank you, Dr. Dagogo Jack. Fat cells, friends or foes? That's the question. No other tissue has been more maligned than adipose tissue. We appreciate that an excess of adipose tissue is frequently associated with an increased risk to develop cardiovascular disease, diabetes, and in fact even cancer. This is despite the fact that our view of the adipocyte has significantly changed since the early 1990s. At that time, we valued the fat cell mostly for its ability to undergo insulin-mediated glucose uptake and to release and re-esterify free fatty acids depending on nutritional status. Now, at this point, we appreciate that the adipocyte is a very complex, metabolically highly active and potent secretory cell. It is capable of releasing a large number of protein products. It is well equipped to manipulate local and systemic lipid levels, and it releases a number of key metabolites. We are going to touch upon each of these emerging themes during the talk today. Obviously, our fat stores are a reflection of the net difference between energy intake and energy expenditure. Energy intake reflecting mostly the uptake of calorically dense foods, while the energy expenditure side reflects the energy required to meet basic energy demands, energy required for physical activity, as well as energy expended on adaptive thermogenesis. We know that the systemic ability to adjust to changing nutrient conditions, a phenomenon that we refer to as metabolic flexibility, very much depends on the ability of the adipocyte and adipose tissue to expand in order to accommodate a potential excess of calories going into the system at any given time. Adipose tissue expansion can occur under a couple of different scenarios, and depending on how effective this expansion process is, it leaves us with either metabolically fit, happy fat cells, or dysfunctional, unhappy fat cells that display a large degree of metabolic dysregulation. Metabolic flexibility is not exclusively a function of absolute fat mass. Plotting fat mass versus the degree of insulin resistance does not reveal a strictly linear relationship. Many of us fall into the category of metabolically unhealthy, obese individuals. That means the more adipose tissue we have, the more metabolically challenged we are. However, some individuals fall into the category of metabolically healthy obese. Despite having relatively high body mass indices, they display normal metabolic parameters, even though they may not be immune to future pathophysiological changes as a consequence of the increased fat mass. The other extreme is lipodystrophy, reflecting the metabolically unhealthy lean individual, displaying severe metabolic dysfunction despite the fact that these individuals have very little, or in some instances almost no, adipose tissue to cope with. Why can some of us maintain normal insulin sensitivity despite being overweight or even obese? And what can go wrong in our adipose tissue as we gain weight? The reasons why an excess of adipose tissue generally causes metabolic stress are certainly complex. However, we can dissect the path to dysfunction into a number of relatively simple steps. An expanding fat pad, just like any other rapidly expanding tissue mass, displays impaired angiogenesis, and that impaired angiogenesis can lead to local hypoxia. In other words, an insufficiency in the local oxygen supply. This then results in fibrosis and the activation of a number of other downstream events, including the unfolded protein response in the secretory pathway. All of these phenomena ultimately trigger increased cellular stress, increased necrosis, and some degree of chronic unresolved inflammation. We can visualize some of these steps in adipose tissue. Increased fibrosis as a result of enhanced production of extracellular matrix components is seen here by trichrome staining and reflected by an enhanced blue stain seen on the right-hand side. This is widely observed in obese adipose tissue from both humans and rodents. We know from both genetic and pharmacological studies that the modulation of the extracellular matrix environment plays a vital role in the health of adipose tissue, both at steady state and during expansion. Elegant studies by the Clement group in Paris and the Smith group in Orlando have underlined the critical implications of many of these pathological changes associated with fibrous adipose tissue. In this context, we have studied the relevance of collagen 6 expression, one of the most abundant extracellular matrix constituents of adipose tissue. We can observe in a growing fat pad that there is a significant reduction in the degree of extracellular matrix accumulation in the context of mice lacking collagen 6. As a result, these mice retain normal metabolic function while challenged by a high-fat diet, despite displaying very large individual fat cells with a reduced incidence of adipocyte cell death. This is apparent when we subject the mice to an oral glucose gavage and observe an improved oral glucose tolerance. Even though these mice have mechanically less stable fat tissue, in the absence of this key extracellular building block, they are metabolically healthier because they can bypass the usual constraints imposed by the fibrous extracellular milieu that the fat pads experience during expansion. Extending the pro-angiogenic component in adipose tissue during expansion can also significantly stave off the fibrotic response, reduce ensuing cell death, and translate into a reduced level of local insulin resistance as a result of reduced inflammation. The accumulation of extracellular matrix is an early event and offers an opportunity for pharmacological intervention with antifibrotic agents. The increased rate of cell death in the context of a disproportionate accumulation of extracellular matrix constituents leads to the attraction of macrophages. These macrophages seen here, surrounding a lipid droplet resulting from a dead adipocyte, start to engulf lipids over the course of several weeks. It is this process that is associated with the chronic inflammatory response. There is no doubt that chronic, unresolved inflammation leads to chronic insulin resistance, a phenomenon that we frequently see epidemiologically. This has also proven to be relevant in a host of rodent models. However, is it invariably a bad thing to have some low level of inflammation at the level of the adipocyte? This is an important question since anti-inflammatory interventions have proven to be of limited benefit with respect to clinical improvements in insulin sensitivity. To probe further into this role of inflammatory responses in adipose tissue physiologically, we took a series of genetic approaches in which we generated mice that have a high level of anti-inflammatory activity exclusively at the level of the adipocyte. We have done this in 3 different ways, shown here. We have expressed the dominant negative TNF-alpha ligand that unproductively engages the TNF receptors. We have also expressed a potent adenoviral anti-inflammatory molecule called RIT, as well as taken advantage of a traditional quote-unquote super repressor of inflammation in the form of an IκB alpha mutant. The specific details of each approach are not critical to understand, but in all 3 cases we have fat cells expressing potent anti-inflammatory factors. We were very surprised to see that in the process of adipose tissue expansion, an inflammatory response is initiated as part of the healthy expansion process. By inhibiting this inflammatory response, we notice that we impair adipogenesis, thereby reducing the number of fat cells available to accommodate any excess lipids, leading to ectopic lipid deposition in the liver. This also led to a compromised intestinal barrier and the release of more endotoxin into the system. So despite the early anti-inflammatory action at the level of the adipocyte, chronic systemic inflammation and severe systemic insulin resistance ensue. As a result, we reached the surprising conclusion that a transient low level of inflammation is beneficial and in fact required for the remodeling of adipose tissue, again highlighting the central role of the extracellular matrix in the process of tissue expansion. There are additional physiological settings beyond mere expansion in which the importance of the extracellular matrix remodeling of adipose tissue is apparent. Fat cells can assume a number of different specialized functions and can in fact be recruited to do so by a number of different interventions. This leads to a surprising complexity in terms of the nature of the different adipocytes in our fat pads. There are classical white adipocytes, as well as brown adipocytes that are specialized in the generation of heat with high expression of the marker uncoupled protein 1. A third cell type, the beige adipocyte, is found in white adipose tissue but has the capacity to develop many of the functional characteristics of brown fat under certain conditions, including cold exposure. An active beige adipocyte is a cell that can convert back to a dormant beige adipocyte that resembles a white adipocyte. All of these tissue transitions critically rely on the remodeling of the extracellular matrix. Let us leave the tissue autonomous effects behind at this point and take a broader view of the adipocyte and its systemic contributions. I want to highlight 3 specific areas in which the adipocyte plays a major role in systemic energy homeostasis. These areas include the adipocyte as a source of protein factors, so-called adipokines, that exert important effects on systemic metabolism. I will focus on adiponectin, an adipokine that my lab has studied extensively. Some of these factors play a major role in lipid homeostasis, and as such, the adipocyte also serves as an important buffer for these critical lipid mediators. I will discuss a specific class of lipids, the sphingolipids. A third area that I would like to mention is the adipocyte as an important source of critical metabolites. In the context of today's discussion, I will focus specifically on uridine. Historically, Flyer and Spiegelman started to look at the adipocyte as a source of enzymes and secreted factors. In the late 1980s and early 1990s, they described components of the complement fixation cascade, anti-inflammatory factor, tumor necrosis factor alpha, as adipocyte-derived product. Our own contributions towards establishing the concept that the adipocyte is a highly, quote-unquote, professional secretory cell expanded the notion that adipocytes release many factors affecting the extracellular matrix, metalloproteases, and also increase the release of pro-inflammatory factors in the obese state. More importantly, the adipocyte produces a series of factors highly enriched in adipose tissue that are released into the system and that have profound effects on systemic metabolism. These factors include proteins such as adiponectin that we first discovered in the 1990s, around the same time that leptin, another key adipokine, was reported by Jeff Friedman's lab. Additional molecules such as resistin, FGF21, and endotrophin, and many others were added to that list of adipocyte-enriched secretory products at later points by us and by many others. For the past 20 years, work in my laboratory has focused on elucidating the physiological function and the systemic impact of adiponectin, the most adipocyte-specific of all secretory factors described to date. This old-fashioned northern blot reflects the first data obtained on this factor. We noticed that adiponectin is highly enriched in fat tissue and dramatically induced in tissue culture over the course of adipogenic differentiation, as seen on the right-hand side. Since this initial northern blot, adiponectin has been a wonderful source of inspiration to learn not only much about the physiological impact of the protein itself, but also much about adipose tissue physiology in general. Despite being exclusively expressed at the level of the fat cell, adiponectin levels in plasma are actually inversely proportional to fat mass, an observation which, at least at first sight, is somewhat counterintuitive, and in contrast to essentially every single other adipocyte-derived secretory protein. We have also learned by comparing metabolically healthy and metabolically unhealthy obese individuals that metabolically healthy individuals display higher levels of adiponectin despite identical body mass indices. This suggests that adiponectin levels more closely correlate with insulin sensitivity rather than merely reflecting fat mass. As we increase our fat mass under normal conditions, adiponectin levels are decreased. We have genetically manipulated mice such that they maintain high adiponectin levels similar to lean mice despite gaining weight, reminiscent of what the metabolically healthy obese individual does. We have done this in rodents on several different occasions, either through a direct overexpression of adiponectin or through the manipulation of other critical cellular pathways, including interventions at the level of the mitochondria. We noticed that under these conditions, the mice dramatically expand their fat mass to become the rodent equivalent of an 800-pound human subject. However, despite being the heaviest mice ever reported, they retain full metabolic flexibility as the subcutaneous fat of these mice effectively absorbs all excess calories, thereby sparing all other tissues from any adverse lipotoxic effects. In fact, the adipose tissue of these mice is a paradigm for healthy adipose tissue expansion. Histologically, comparing these metabolically healthy mice to their leaner, unhealthy counterparts, we notice that the metabolically healthy mice have many more but smaller fat cells with widespread adipocyte hyperplasia. They do not display the conventional elevated levels of inflammatory markers as judged by the reduced number of immune cells infiltrating the tissue. They are better vascularized and show a reduced degree of fibrosis. Healthy fat pads translate into insulin-sensitive livers that have less steatosis, and better cell function is also spared to maintain fully functional islets. Positive changes in adiponectin levels are also a reflection of therapeutic improvements in systemic insulin sensitivity. For example, upon exposure to a PPO gamma agonist, we can see that the relative increase in adiponectin is proportional to the effectiveness of the drug to improve systemic insulin sensitivity. In fact, in the absence of adiponectin, PPO gamma agonists reduce their potency to induce improvements in insulin sensitivity. This type of correlation has been seen in dozens of different studies with distinct classes of anti-diabetic drugs. So we conclude that adiponectin levels are excellent integrators of adipose tissue health and systemic metabolic flexibility. Most importantly, changes in adiponectin levels induced by either weight loss or pharmacological interventions reflect improvements in insulin sensitivity. As a result, many laboratories have studied the effects of adiponectin on a large number of different target cells and tissues. There is not a single tissue in the system that is not directly or minimally indirectly affected by adiponectin's actions. In many instances, improvements at the level of the tissue are not merely a correlation to increases in adiponectin levels, but rather they are a direct consequence of it. In order to analyze the function of adiponectin in different target tissues, we have constructed a series of models that allow us to challenge specific cell types in target tissues with a pro-apoptotic stimulus. We refer to these mice as the attack mice for apoptosis through triggered activation of caspase 8. These mice have a transgenic cassette encoding a suicide gene that we can specifically activate in the respective target cell. In this way, we can probe for the ability of the specific cell type to resist cell death as a function of adiponectin concentrations in the system. Once we have ablated the target cell population and stopped the apoptotic stimulus, we can also use the same system to test the capacity of the tissue to regenerate as a function of adiponectin concentrations in plasma. We have done this for a number of different tissues, including the heart, the pancreatic beta cell, the podocyte, the kidney cell, critically affected by diabetes, as well as for many other tissues. For instance, in the cell ablation model for the cardiomyocyte, the so-called heart attack mouse, we activate the suicide gene and monitor the number of animals that survive as a function of time post-initiation of the treatment, and we can see that the higher the adiponectin levels, the higher the survival rate. Then we focus on the pancreatic beta cell, highlighted here in wild-type islets with a green insulin stain. In this case, we use the pancreatic islet cell attack mice, a system that we refer to as the panic attack mice. We can see that post-ablation of the beta cell, we have islets which almost exclusively contain alpha cells, as judged by the red anti-glucagon stain, and contain very few remaining insulin-positive green cells. In the absence of adiponectin, these beta cells are a lot more susceptible to the pro-apoptotic stimulus, as judged by the fact that we can monitor the systemic blood glucose levels and see that the glucose levels are higher, reflecting reduced beta cell viability and increased loss of beta cell mass in the absence of adiponectin. And then, if we look at the regenerating beta cell mass, once we uniformly ablate insulin-positive cells in all genotypes to the same extent, we notice that the presence of increased levels of adiponectin in the system significantly improves the ability of the pancreas to regenerate beta cell mass and to reach near euglycemic conditions, an observation with profound implications for both type 1 and type 2 diabetes. Similarly, in the kidney, we can induce widespread elimination of the podocyte in the pod attack mouse, and notice that with elevated adiponectin in the system, we improve the functional recovery of the kidney dramatically, reflecting the profound impact of adiponectin on improvements in kidney function in the context of chronic kidney disease and diabetic nephropathy. So, with adiponectin, we are looking at the protein that has potent anti-apoptotic function in a number of different cell types. In addition, it can be pro-angiogenic, making sure that the expanding fat pads get appropriately vascularized. It can be anti-atherogenic, with important implications for cardiovascular disease. It is a potent anti-inflammatory factor. And it can stimulate the generation of new small fat cells, so it's pro-adipogenic. It can cause potent improvements at the level of hepatic insulin sensitivity, and it exerts positive effects on wound healing. Adiponectin has given us many additional insights over the years. Adiponectin is one of the best biomarkers for existing metabolic disease. Low levels of adiponectin reflect high degrees of insulin resistance. It is one of the best prospective biomarkers for future incidence of cardiovascular disease and diabetes. Its genetic dissection has provided us with a lot of valuable research tools. Mutations in the adiponectin gene in humans that lead to reduced adiponectin levels are closely tied to a metabolic syndrome phenotype. At the preclinical level, the adiponectin promoter cassette has allowed us to highly specifically manipulate the mature adipocyte and to generate a number of genetic tools, allowing us to monitor the generation of new fat cells under many different conditions. And importantly, the analysis of adiponectin's physiological impact has highlighted a number of new therapeutic areas. There is widespread interest in the adiponectin receptor as a drug target. A lot of effort currently goes into the identification of adiponectin receptor agonists that bear promise as therapeutic insulin sensitizers. These efforts should be further facilitated by the availability of high-resolution structures for the adiponectin receptors that were recently published by Takashi Karawaki's group in Japan. Both the Karawaki group in Tokyo and the Matsuzawa group in Osaka have contributed significantly to our understanding of adiponectin physiology over many, many years. With the concerted efforts amongst our three groups and many others in academia and industry, I am confident that there is ample room for the development of a clinically relevant class of adiponectin receptor agonists for the treatment of metabolic disease. In addition to all of these aspects, adiponectin has also taught us much about the ability of the fat cell to manipulate lipid factors. The connection between adiponectin and lipid metabolism is particularly apparent when we look at the broad category of lipids referred to as the ceramides. The ceramide subspecies of sphingolipids have been linked to inflammation, cell death, and insulin resistance. Ceramides can be degraded to generate sphingosine